second category is prokaryotic cells. They don't have a nucleus or membrane-enclosed organelles. They do have genetic material, but it's not contained within a nucleus. Prokaryotic cells are always one-celled or unicellular organisms, such as bacteria. So, what are organelles? Organelle means little organ. Organelles are the specialized parts of a cell that have unique jobs to perform. Let's start with the nucleus, the control center of the cell. The nucleus contains DNA, or genetic material. DNA dictates what the cell is going to do and how it's going to do it. Chromatin is the tangled, spread-out form of DNA found inside the nuclear membrane. When a cell is ready to divide, DNA condenses into structures known as chromosomes. The nucleus also contains a nucleolus, which is a structure where ribosomes are made. After ribosomes leave the nucleus, they will have the important job of synthesizing or making proteins. Outside the nucleus, the ribosomes and the rest of the organelles float around in cytoplasm, which is the jelly-like substance. Ribosomes may wander freely within the cytoplasm or attach to the endoplasmic reticulum, sometimes abbreviated as ER. There are two types of ER. Rough ER has ribosomes attached to it, and smooth ER doesn't have ribosomes attached to it. The endoplasmic reticulum is a membrane-enclosed passageway for transporting materials, such as the proteins synthesized by ribosomes. Proteins and other materials emerge from the endoplasmic reticulum in small vesicles, where the Golgi apparatus, sometimes called the Golgi body, receives them. As proteins move through the Golgi body, they're customized into forms that the cell can use. The Golgi body does this by folding the proteins into usable shapes, or adding other materials onto them, such as lipids or carbohydrates. Vacuoles are sac-like structures that store different materials. Here, in this plant cell, the central vacuole stores water. Going back to the animal cell, you will see an organelle called a lysosome. Lysosomes are the garbage collectors that take in damaged or worn-out cell parts. They are filled with enzymes that break down this cellular debris. The mitochondrion is an organelle that is the powerhouse for both animal and plant cells. During a process called cellular respiration, the mitochondria make ATP molecules that provide the energy for all of the cell's activities. Cells that need more energy have more mitochondria. Meanwhile, the cell maintains its shape through a cytoskeleton. The cytoskeleton includes the thread-like microfilaments, which are made of protein, and microtubules, which are thin, hollow tubes. Some organisms, such as plants that are photoautotrophic, meaning they capture sunlight for energy, have cells with an organelle called a chloroplast. The chloroplast is where photosynthesis happens. It's green because it has a green pigment called chlorophyll. Plant cells also have a cell wall outside of their cell membranes that shape, support, and protect the plant cell. Animal cells never have a cell wall. There are many other unique structures that only some cells have. Here are just a few. In humans, for example, the respiratory tract is lined with cells that have cilia. These are microscopic hair-like projections that can move in waves. This feature helps trap inhaled particles in the air and expels them when you cough. Another unique feature in some cells is flagella. Some bacteria have flagella. A flagellum is like a little tail that can help a cell move or propel itself. The only human cell that has a flagellum is a sperm cell. In summary, remember, eukaryotic cells are plant and animal cells with a nucleus and membrane-enclosed organelles, while prokaryotic cells are unicellular organisms without these things. All cells have a cell membrane, cytoplasm, and genetic material. And even though only plant cells have chloroplasts, both plant and animal cells have mitochondria.
So, good morning, everybody. Can you hear me? Can you see me?
Yes, both.
Thank you. Welcome to our course. So, my name is Lukas Huber. I'm a cell biologist working at the Medical University of Innsbruck, and it's my pleasure to give you this course today. Unfortunately, it has to be online, but I will try my best to make it as good as possible for you and as entertaining and as interesting as it could be as an online lecture. Now, the video that I have shown to you at the beginning is maybe a bit too naive for the knowledge base that you guys have meanwhile obtained during your studies. But nevertheless, it summarizes visually everything what we are interested today. You saw prokaryotic cells, eukaryotic cells, you saw animal cells, human cells, specialized cells, plant cells, and all those cells contain organelles, and those organelles are in the focus of our investigations. So I give you some examples. I'm working now mainly in the field of the cell biology of diseases, especially rare diseases. Now, these diseases affect the function usually of one or more organelles. For instance, mitochondrial diseases, these are all genetic diseases. Most of them are monogenetic diseases. They affect protein secretion, protein transport, protein processing. They affect lysosomes, like lysosomal storage diseases. And by telling you that, it becomes obvious that one has to analyze these organelles. Now, these organelles can be analyzed by purifying them. And this is what we are going to discuss today. And I will give you a course on how you do that. I have pepped up the course with videos, and I have also videos from the bench where I can show you different steps of this purification procedure, and you can learn with me how to use that with simple devices on the bench. I have distributed literature to you where protocols are given. And my goal in this course is to bring you that far that you understand these protocols and that you get encouraged to use, to make use of the knowledge that you obtain today. Now, some organizational remarks. So we are a small group. At the moment, there are 10 students online with us today. And I encourage you to actively participate in the lecture by asking me questions. If something is unclear, or if I should go into more detail with something, or if I should repeat something, or simply if you would like to have an explanation, why is it like that? For that, you can use the chat room. You can take the microphone and ask your question, and it would be, of course, extremely nice when you use the microphone that you turn on the video, that I can see your face. But I also acknowledge and accept if you would like to avoid that and not use the video. But it makes it usually for the teacher as well as for the students a bit more pleasant to get into contact. And that is what I would like to achieve also, to get into contact with you. Because when you have finished that course, you should be encouraged to contact me later when you need help, when you need advice from a cell biologist, when you have questions, because the course is laid out for you in order to make use of the knowledge during your master thesis and also later during your PhD thesis. And I will give you examples during the course why that will be the case. Now, we will be busy for several hours today, maybe 3 hours. We will make breaks in between for having a coffee and then to proceed again with a refreshed mind. And in order to get this course into your study curriculum and you get a mark, you have to write a homework, an essay. And how that has to be done, we will discuss at the end of the course, but it's all doable. It's not a tremendous amount of work, but it's more like a reflection of what you have heard. And we will talk about that in more detail. So do you have now, before we go into the scientific content, any questions for me? Is everything clear so far, or would you like to make a suggestion or ask something? Okay, if that is not the case, yes.
Sorry, I have a quick question. Oh, sorry.
Hello, Andrea. Good morning.
Good morning. You said that you provided some materials.
Yes.
And I was not really able to find them. Should they have been in the email that
so in the email you have received, there was a WeTransfer link that you just have to double click on that link, and it was written a sentence that you can find that under the following link. And when you click on that link, a WeTransfer dialog box will open and you can download it on your computer.
Okay, thank you.
On top of that, I have pasted into the chat room for you a PDF of the slides that you can download. Have you seen that I have sent at the beginning? You have to look into the chat room.
Oh, okay.
Yeah, and there is a PDF file, Subcellular Fractionation, summer semester 2022. And when you click on that, you can directly download it for the chat room function.
I still can't see it. Maybe my connection was established afterwards. Would you be able to send it again?
Yeah, in one of the breaks, I will send it again.
Okay, thank you.
Okay? Yeah. Then we make a break every 45, 50 minutes, and then in one of the breaks, I will send it to you.
Thank you.
Thank you. Are there more questions at this point? Okay. Then let's proceed. So why are we interested in looking at these organelles? You know, these organelles are composed of proteins and lipids. And so they have a lipidome, they have a proteome. Some organelles have a genome as well, like nucleus or the mitochondria, for instance. And the human proteome, so the protein content of an entire cell, of an entire organism, is a huge entity. What does that mean? You know, when we follow the hypothesis of one gene equalizes one protein, there should at least be a huge number of proteins non-modified canonical proteins coded by the genome, and that should be around 20,000. But it's not one gene equalizes one protein. When we take into account products of splicing, alternative splicing, then the ones containing single amino acid polymorphisms, which arise from non-synonymous single nucleotide polymorphisms, and post-translational modifications. And just for your interest, there are around 200 of them described, protein modifications that occur post-translationally. Then we can have as many as 100 different proteins that can potentially be produced from a single cell. So we have 100,000s of protein species in a cell. And this is a tremendous amount of proteins that is present in the cell. Now, that makes the human proteome extremely complex. When we take, for instance, a body fluid like blood plasma, this is already a very complex mixture. And what the problem in there is that it has a tremendous dynamic range of protein concentrations, like in a cell, varying by 10 orders of magnitude. And this is also reflected in the protein concentration range of tissues, for instance, or cell lines with 7 orders of magnitude. So that means we have a tremendous challenge technically with all high-end instruments that we are using, pre-fractionation techniques, mass spectrometry, gel-based techniques, detecting the low and ultra-low abundant species of proteins with concentrations from smaller than 10 to minus 12 molar in the presence of very high copy number protein molecules into concentrations from more than 10 to minus 6 molar. So I'll give you an example. Actin, keratins, structural proteins in the cell are extremely abundant, whereas signaling molecules like small GTP binding proteins, for instance, like the oncogene RAS, are extremely low abundant. So you have a few thousand copy numbers of this oncogene in the cell, and you have millions of copies of other proteins. And the problem is then, how do you detect them? Because with conventional techniques, you would not see them. They are masked by the huge amount of the other proteins. So we need technologies to see them. And one of these technologies that has revolutionized the way how we look at cells, how we analyze proteomes, lipidomes, has been the introduction of subcellular fractionation, which is not a new technique. It's a rather old technique that has been established already by the pioneers of cell biology, like the ones that have worked on many years on the Golgi complex, like George Baladi, who received the Nobel Prize for that. They have purified already in the end of the 60s and beginning of the 70s, Golgi stacks to quite pure stacks that you could analyze biochemically at those early days in cell biology, like Christian de Duve, who has discovered the lysosome and endosomes and received the Nobel Prize for that. They have been able to purify those organelles. And those technologies have now been revitalized and picked up by cell biologists in order to use them for other complex questions. Many want to get access to the knowledge of intracellular organelles, how they are composed, and to look even at multi-protein complexes at large protein machineries in the cell. And there are many large machineries in the cell. I would like to remind you the ribosome, for instance, or the proteasome that degrades proteins, or the anaphase promoting complex, just to name a few. So proteins usually don't exist in isolation. They exist in their social context where they interact with other proteins and where they can perform their magnitude of functions only within the framework of their social context. And usually they are associated with organelles And these organelles have particular functions then, depending on whether these protein complexes associate in the right manner with them or not. So that sets a bit the stage for you in order to understand why we apply subcellular fractionation, because this technology enriches low abundant proteins and signaling complexes. All of a sudden, when we can enrich samples for certain organelles, we start seeing proteins that we have never seen before, like this picture that I've pasted here on the right of this iceberg. You know, we only see the tip of this mountain in the sea, and there is a huge amount underneath that is not visible to us at the moment without those technologies. That means subcellular fractionation techniques very efficiently reduce sample complexity. The technology itself is a rather flexible and adjustable one that you can adjust to your personal needs, as you will see. The protocols that I will introduce to you can be adapted basically to purify every single organelle that you have seen in the introduction video before. You can combine it very efficiently with downstream functional analysis. Example given, you can purify organelles like the mitochondria. And once you have purified mitochondria of a patient, so to say, from patient cells, from patient fibroblasts that you can obtain with a biopsy, like we do it routinely, the clinical setup with our patients of rare diseases, rare muscle diseases, for instance, we can isolate the mitochondria and we can measure them. We can analyze them for their protein content. We can measure the membrane potential. We can measure the redox potential. We can look in detail at these organelles and study them functionally, as well as from the point of view of their composition. And those technologies are also compatible with every downstream biochemical analysis, with two-dimensional analysis on gels or gel-independent techniques, mass spectrometry techniques, just to name a few of them. However, there is, of course, a word of caution that has to be raised here, and that is that subcellular fractionation remains a major bottleneck. And we have these organelles composed of proteins and of lipids, and each organelle has a bit of a different composition. And I'm showing you here an electron micrograph, a beautiful electron micrograph from a textbook from a plant cell. You see the cell wall, you see the cell membrane, and you see inside these organelles, you see the different Golgi stacks here, like this staple of pancakes. You see membranes of endoplasmic reticulum. You see lysosomes, multivesicular bodies here with different content. You see mitochondria as well, and you see free ribosomes in the cytoplasm. Everything is very densely packed. And they all have a different protein and lipid composition. However, they share similar physical properties, although they look very different in their morphological appearance. And these similar physical properties, they are defined by protein to lipid ratios, by the nature of the lipids, and by the netto charges they contain. And there are, of course, differences between tissue cultures, cells, and real tissues. And just to explain to you before we start looking into more detail, it's very surprising, but it's a fact that it is much easier to isolate organelles out of tissue than it is out of tissue culture cells. And there is a reason for that that I will explain you a bit later. Now, as I said, they look remarkably different, these organelles, when we look at them with microscopy. But they have, unfortunately, similar properties, physical properties. And most of the separation techniques, or many of them, are based simply on their physical properties. That means based on their physical properties, we can separate them. because they have a different specific weight, or they migrate different ingredients, or they have a different netto charge composition on their surface. Now, we will therefore cover in this lecture today these chapters that are listed here. First, we will briefly talk about this membrane transport and organelles and focus on one particular organelle class for didactic reasons. And with this particular organelle class, we will play through several options of how you can separate and purify these organelles. However, everything I'm teaching you today can be applied to all other organelles as well, with minor changes into protocols that you will understand then later. However, what is the prerequisite in order to get access to these organelles, that we can purify them in first instance at all, is that we need to homogenize the cells first. And then we separate the organelles. But this step, the homogenization, is the ground, the basis, so to say, of every other purification procedure that is downstream of that. And therefore, we will devote very much attention to the homogenization and to the pitfalls in the homogenization and why you have to take care about certain rules here. Once you have homogenized your sample and you have a solution where these organelles freely swim after breaking up the cells, you can try to pick those organelles out and to separate them, either by centrifugation steps like density gradients, density shifts, by electrophoresis technologies that take advantage of the different charges those organelles might have, or by selective fishing approaches by using antibodies, an approach that we call immunoisolation, or by technologies where you can introduce fluorescent markers into organelles and use fluorescence activated organelle sorting, which is based on flow cytometry technology. Now, the fractionation of the cells. So the fractionation of the cells is the breaking open and then the subsequent separation. So you put the cells in different parts and this usually then when you want to purify them requires a significant number of cells, that you get sufficient amounts of these organelles. And you can break open the cells. So physically, you can lyse the cells biochemically, and you can homogenize the cells. Now, how do you disrupt these cells? Chemically, you have several options for disruption. You can take alkaline conditions, you can take organic solvents that dissolve the lipids, you can take detergents to solubilize membranes. There are enzymatic activities as well that you can apply, like lysozymes or chitinase to digest away the cell wall, so to say. There are physical treatments. You can apply an osmotic shock on the cells. So when you change the osmolarity of the surrounding medium, you can put the cells under osmotic pressure where they easily disrupt them. You can freeze the cells and thaw them immediately after that. That also will disrupt the membranes and you get access to the cytoplasmic space here. There are mechanical options like sonication, homogenization with rather simple devices like syringes I will show you with different needle sizes. There is, for large amounts, the good old French press that maybe some of you might have used already. I will explain the principle then later, which is something you can use also for bacteria to break them up when you use bacteria as protein factories, so to say, and syringes with needles. Now, chemical disruptions. Chemical disruptions are based on the use of detergents. You know, detergents are something like soaps. And depending on the concentration where to use them, they form micelles, and you are working below the critical micellar concentration so that you can solubilize the membranes and free the proteins. And the trick thereby is that you, for instance, only get rid of the plasma membrane, but leave the organelles intracellularly intact, that you use mild detergents to generate, basically like shown in this insert picture here, holes into the plasma membrane. The detergents of choice here are tritones like Triton X100 or Nonitet P40. They can permeabilize cells by solubilizing the membranes. Detergents can be expensive. They are highly purified reagents. They have, of course, the disadvantage that they can also denature proteins. And you have to disrupt them, to remove them after the disruption. And for some of the downstream analysis, like mass spectrometry, you also have to remove them because they would disturb the performance of the machines.
I have a question.
Yes, please.
Why is it crucial that the detergent is non-ionic?
It's the non-ionic detergents, typical detergents that you utilize to disrupt the plasma membrane and to solubilize the plasma membrane. Other detergents like twitterionic detergents you can also take, but they usually disrupt cells in an asymmetric manner. So, for instance, twitterionic detergents make holes preferably in cholesterol-rich regions of the plasma membrane, like caveolae or lipid rafts, and other detergents like Triton X114 you use typically to solubilize transmembrane proteins into the soluble fraction, go the peripheral membrane proteins, and the integral, the transmembrane, remain in the detergent insoluble fraction. This is empirically found out.
Okay, thank you very much.
Yes, thank you for the question. So this is the French press. So basically the French press is a closed metal container which has a hole somewhere on the bottom. And you apply pressure either manually by a wheel that you turn down that increases the pressure in this sealed vessel, or by hydraulic pressure. And the idea is that when you insert this piston and press it down, that you increase the pressure. And in order to escape the pressure, the cells are pressed through this little hole, for a small hole on the bottom. And by squeezing through that hole, you disrupt the cells. And the content of the cells basically is spilled out. So you have then underneath, after the forcement of the cells through that small hole, you have underneath coming out the nucleus, the membranes, the plasma membrane disrupted and vesiculated, the mitochondria, all the other organelles, and the cytoplasm. But the main use of the French press is for prokaryotic cells, because bacteria you can press through very easily, you can disrupt them, and then you can harvest, for instance, the proteins in the supernatant that you have expressed in the bacteria. A bit more sophisticated and more gentle is already a sonicator. You know, also sonicators are found at every institute to sonicate cells, to sonicate DNA, to sonicate liquids in order to make emulsions or something like that. And the sonicator tip is shown here. This is the tip, and those tips you can purchase in different sizes according to the amount of cells you would like to break up. You have larger tips or smaller tips that they fit also into the tubes, and they are immersed directly into a cell suspension. And once you turn on the sonicator and it starts vibrating, the high frequency sound waves start then disrupting the cells. And of course, here that has also to be empirically tested, but all these details are then usually given in the protocols, and it is written there, you should use this kind of sonicator tip with that kind of size. You should use this kind of time. But what is always important is to keep in mind already, here is perform all your steps in a cold environment on ice, because that is very important, as you will see later. Now, then we have the homogenization with so-called down cells. Now, the cells are placed here in a closed vessel. Usually, this is made out of glass. And in that vessel, a tight-fitting plunger is inserted. And this plunger rotates and is pulled up and down. And during these two movements, the rotation around the own axis and the pulling it up and down, the cells are disrupted by squeezing them through the narrow space that is left between the plunger and the wall, the glass wall, and the vessel wall. And these systems, they are manufactured by special companies that are specialized on scientific glassware that is used in laboratories. They are usually not very expensive, but they can be designed that you can buy plunger and vessel systems with exactly defined distances to the wall in the micrometer space. And that means you can adjust the space width here, which helps you to deal with larger cells or with smaller cells that you can then very easily disrupt with this technology. And this shows you some examples of these plungers, that they have different shapes, different sizes. You can connect them to an electric motor device that they rotate with kind of high speed, and then you can pull them up and down. You can cool that, put that into an ice bucket, and that is a very convenient way of homogenizing cells. What is a very smart and easy way is the homogenization by using a syringe and a needle. And one of my goals is to show you techniques that you can easily apply yourself in the experimental setup where you will be exposed to in the future. And what you find in every laboratory is, for instance, a needle. Now, this is for an example of a 1 milliliter syringe, and it has an orange needle. So these syringes can be exchanged to 2 milliliter syringes, 5, 10, up to 20. The needles are color-coded, and the color code means they have a certain size in the diameter of the opening of the needle. And this is given in gauge, in 20 gauge, 22 gauge, 21 gauge, 24 gauge. And this is the measure. And usually in the protocols, it's given you should take a needle with a blue color, orange color, black color, white color, and this depends on the opening size. Now, with such a needle, you can aspirate, you can fill in here the suspension where your cells are suspended, and then you can press that through the needle several times into a vessel. And every time you press it through and you suck it in again, you apply shear forces to the cells when they are pressed through the needle, and by that the cells can be disrupted. And the organelles and the nuclei can be released, and the sizes you can vary. Now, before you homogenize your cells, you have to spend some thoughts that you have to consider. And that is, of course, an old principle in biochemical analysis. You know, the old biochemists who purified proteins out of cell lysates with huge columns, with chromatography approaches, used to say, shit in, shit out. You know, when you have shit that you put into your column fractionation, a bad sample, so to say, of bad quality, you get shit out at the end of the day, you get damaged proteins. What that means for you is that you have to take care about the quality of the cells, because certain cell types are very sensitive to the number of passages that they are exposed to during their life. And this directly correlates with the quality of the homogenization results. So with other words, you know, cells that are passaged over long periods of time, they turn on an aging program. And when cells go into senescence and they age, they change their shape. And cells that had a size of 20, 30 micrometers in diameter and were very nicely round or cubic form on the tissue culture plate, they change over time and they flatten out, they cover a huge surface, they rearrange their cytoskeleton, and they become very, very unpredictable in their behavior when you try to homogenize cells. Therefore, use always cells with a known passage number. So usually when you come to a lab and you start your thesis work there, you work with a certain cell line or with a certain strain, you should ask it, which passage number they have, how long they're in a culture. You should insist on getting an early passage, that you split yourself, you cultivate yourself, that you document how many passages the cells had. Because usually when those cells age over time, and you always use them for your assays, the assays show varying results over time. Biochemical assays, immunofluorescence assays, electron microscopy assays, everything you are applying to the cells changes when the cells undergo senescence. And what is also very important is that you document that carefully in your laboratory protocols. And just keep in mind that your supervisors can only help you finding out problems from when an experiment has not worked anymore, when you have a good documentation, when you have written down the date, when you seeded the cells, how many cells you have seeded, how the cells looked like. When you make your little remarks in your notebook, then it's much, much easier for you and your supervisor to trace back where potential pitfalls might have arisen from. What is also important is the confluency of the cells on the plate. This is a critical parameter because the cells, when they grow on a tissue culture plate, usually they grow as long as they have free space. And once the space is occupied and they start touching each other, like my two hands do it now, they stop dividing and they make cell-cell contacts. And this is what we call contact inhibition. However, not all cells do have contact inhibition. Tumor cells classically don't have contact inhibition, and then they start growing on top of each other. What is important for you to know is once they have made contacts, they are easier to homogenize for you. And this comes back to my remark that I made a few slides before, that it is easier to break up tissue than it is to break up round cells. Why is it like that? Imagine you have a sheet of cells that is tightly connected to each other by junctional complexes, lateral cell junctions, desmosomes. They translate their junctional contacts into the interaction with the cytoskeleton in the cell, and the three-dimensional network is then generated in these tissue culture plates where these cells form a sheet. Now, this sheet, once you get it off from the tissue culture plate and you put it in your syringe, for example, requires only very little shear forces to break it up, because the shear forces you apply are multiplicated and translated into the cell through the cell-cell contacts and the cytoskeleton that links these cell contacts with each other. And then you can very gently break up the cells. And this is important because when you apply too much shear forces to the cells, you break up organelles which you would not like to break up. And I give you two examples. One example is the nucleus. Now, the nucleus contains the DNA. The DNA, and that you know from your practical courses, is a slimy piece, you know, when you purify and isolate it. It's like a piece of slime. And you can imagine when you over-homogenize your cells and you break up the nucleus and the DNA starts leaking out of the nucleus, this little slimy piece of genetic information will, in a very unpredictable way, trap organelles because it simply glues everything together. Cytoskeleton parts, membrane parts, organelles are in a very unreproducible way accumulated in this protein sink, so to say, that you generate. And this should be avoided. Second example, the lysosome. You know, there is a reason why nature and evolution has put digestive enzymes like proteases, lipases, and all those lytic enzymes into a bag protected from the cytoplasmic space. Because in the case those enzymes would be freely available in the cytoplasm, they would digest the cell away. And this is not what a cell wants. You know, it wants to have this digestive enzymes somewhere on a safe place where they are kept away from causing harm to the cell. But everything you introduce into that digestive bag by protein transport, by vesicles that go there and fuse with it, they can release the content into this digestive bag. And in this protective environment, digestion can take place. And the amino acids can then be reutilized, transported out of the lysosome again for protein synthesis. The lipids can be reutilized. Now, coming back to our problem, when you homogenize the cells, you can easily break lysosomes when you are not careful, and then these digestive enzymes leak out. So that means the confluence of the cells is a very important parameter for you before you start. And then a very easily understandable note from my side, the technical note is, be careful when you transfer your plates into the incubator, because you know, usually you seed your cells after trypsinization in fresh medium with a pipette on the plate, and then carefully transfer this plate back into the incubator and make sure that it really sits flat on the rack in the incubator and is not tilted or touching another plate when you don't carefully put it in. And that can easily happen when you passage dozens of plates during the day, because then the cells would accumulate on one side and would not cover the entire surface. So just simple tricks, take care about the quality of your cells. And then all these steps have to be performed on ice. And this shows you a typical bench space out of my lab, for instance, where we have these little buckets, isolator buckets, but you can use a simple styrofoam bucket as well that is left over from purchase deliveries to your lab, from chemicals or biologicals that arrived in the lab. Usually many of these styrofoam containers are available. And you fill them with crushed ice. And all these steps have to be performed on ice. The buffers have to be pre-cooled that you use on ice. The pipettes, like this one-way Pasteur pipettes out of plastic. The cells have to be pre-cooled. Here you see a plate, a 10 centimeter plate of cells with medium in it. Usually you can put a metal plate where then the cells flatly sit on top of that. So why do we work on ice? This is very crucial, because when you transfer the cells to very low temperatures, so that means when you chill them down to ice temperatures, metabolism in the cells is stopped, protein transport, vesicle transport is stopped, all metabolic activities are extremely slowed down. Those are important features when you study protein transport and sorting, for instance, because you want to generate a snapshot of the organelle composition, for instance, after certain treatments where you want to freeze down the cells that no further protein transport nor change in the organelle proteome, in the lipidome takes place, like you would take a snapshot with a camera of the cells, a biochemical snapshot. At the same time, you reduce the activity of your enemies. And the enemies are phosphatases, proteases, lipases in the cells that are set free during the homogenization procedure, or like phosphatases that diffuse freely throughout the cell. Because you want to keep, for instance, the natural protein phosphorylation state that you want to analyze later of the organelle-associated proteins or plasma membrane-associated proteins that you would like to analyze. And all those enzymes are slowed down in their activity when you put the cells on ice. On top of that, every buffer that you use in the homogenization and later in the purification should be supplemented with inhibitor cocktails against proteases. Now, these cocktails can be purchased from laboratory suppliers, from chemical suppliers, and usually you add routinely such a cocktail to your buffers in order to inhibit the proteases and the lipases. Now, once you have your plate here on the ice.
Sorry.
Yeah.
I have a question.
Yeah, please.
Why is it not important that the Pasteur pipettes in this picture touch the ice? Shouldn't they be sterile?
No, at that stage, they don't need to be sterile anymore because these cells you do not transfer back anymore to the incubator.
Okay, so they're just here for homogenization.
They are here for homogenization, for exchanging the buffer, for washing the cells. The reason why I'm using these plastic Pasteur pipettes, for instance, is they have a relatively large outlet, a larger outlet than the glass Pasteur pipettes. And by that, the pressure you put on the cell layer during the washing is much lower, and you don't spray off basically the cells of the tissue culture plate. However, there are situations, you're absolutely right, when we do pulse chase experiments, for instance, where you want, after exchanging the buffers or binding something at the surface at 4 degrees of the cells, to shift this culture plate back into the incubator at 37 degrees in order to allow internalization, for instance, then you would use sterile pipettes. Okay?
Okay, thank you.
So once you have the cells here, and I will show you then a video where all these steps are summarized and where you see how that works in real life on the bench, you exchange the medium, and now you want to get the cells off. Yeah. So for getting the cells off the plate, because they make contacts, you know, these cells secrete matrix protein on the top of the plastic. And once the matrix proteins are secreted there, they form a layer like collagens, laminins, and so on. And then the cells express receptors where they can bind to the matrix that they secrete by themselves. So they usually sit rather tightly fixed on these plates. Sometimes the plates are coated with fibronectin or with charges. So you need to get the cells off. You can get them off enzymatically by using trypsin, because trypsin is something you use for splitting the cells. But when you use trypsin, the disadvantage is that the cells round up. Yeah. I don't know if you have observed your cells when you split them, when you passage them. You have seen that already in a phase contrast microscope, that the cells, after a few minutes being exposed to the trypsin solution in medium, they change their shape. They become round, they round up. And once they are round, they float off from the surface into supernatant, and then you shake a bit the dish, and all the cells float off. And then you have all the cells in the medium, and you can take this medium, you centrifuge the cells, and you give them fresh medium, and then you split them. From everything I've said to you before, that it is easier to homogenize tissues or cells that have tight contacts when they come in sheets, it becomes obvious that trypsinization is not the best choice, because round cells are very difficult to homogenize. A round cell slips through this homogenization procedure much more easily than a sheet of cells does. And that means you need to apply more homogenization forces, more shear forces. The more shear forces generate more heat, which is dangerous for the sample. Oxidation of the sample can take place, protein digestion can take place. And on top of that, the homogenization becomes much more harsher, and you run into the danger to break lysosomes or to break the nucleus. Therefore, a method of choice here is using a simple scraper to scrape the cells off. Now, these scrapers are used to collect the cells, and they come down in little sheets. And usually, these scrapers, they have a Teflon, a piece of Teflon, where you can scrape the cells in a very gentle manner off the blade. These scrapers exist in different flavors, in different sizes, with different surfaces. This is something made by our workshop, for instance, something that we prefer to use because this Teflon piece you can easily cut out of a piece of Teflon and put it into this metal holder rather easily. And then you can scrape the cells off, but you can buy plastic scrapers, even sterile scrapers. They are very cheap and can be purchased everywhere. And when you scrape the cells off here, they come off in a form of a little sheet like skin. And it looks a bit like, you know, everybody of us has had a sunburn in his life already. And after having a sunburn, the dead cells of the epidermis, they come off like little sheets. And that is what the cells look like. And you can release them by that very gently from the plate and can then homogenize them. And then after scraping the cells off completely, then you collect that liquid and put it into a tube, and then the first centrifugation is taking place for the rebuffering. And then you can homogenize them by a passage through a needle or through the tip of a pipette. And the first fraction you generate by that is the so-called post-nuclear supernatant. Why is that called like that? Because after breaking up the cells and generating this solution where the cells have been swimming freely in there, but they are open now and release their content and vesiculate, you do apply a gentle step of centrifugation, and you centrifuge down the nuclei. That's in every protocol the same. This is the first step to generate the post-nuclear supernatant. So when you are working in a cell biology lab that is interested in lysosomal organelles, Golgi membranes, endoplasmic reticulum, you proceed then with the further purification with this post-nuclear supernatant. However, in case you are working in a laboratory that is interested in protein transport into the nucleus or into the architecture of the nucleus or in transcription factor research, you want to proceed with the nuclear pellet and you would discard the post-nuclear supernatant. So that means already after this first homogenization and this simple first separation, you can achieve a separation into nuclear fraction and a membrane fraction containing the rest of the cell, all organelles, all the cytoplasmic content, the large protein machineries, as well as lysosomes, mitochondria, and everything else. One point is extremely important here, and is to mention that you should monitor the homogenization process. Usually in the protocol is given, you transfer the scraped off cell solution into this syringe, and then you apply 5 to 8 different strokes with the syringe with a needle size of given 22 gauge or something like that. However, every cell is different when it's grown in different laboratory conditions, and it is wise to monitor this step by a simple microscopy step. You know, in every lab you find this kind of slides, the glass slides you use for microscopy for histology. Just take from the needle, after 5 to 6 strokes of the needle, a droplet, or put it on a glass slide, put a cover slip on it, and go to your tissue culture microscope and have a look at it. And then you will see unbroken cells. You will see broken cells where nice nuclei are visible. You will see a lot of organelles floating around. All that you see in rather low magnifications already under microscope. And make it as a rule of thumb that you stop the homogenization when you have homogenized around 80% of the cells. So when the amount of unbroken cells is 20% or something like that. And because when you over-homogenize the cells, you will see the nuclei breaking up and the DNA leaking out at the end of the day. And this is this homogenization that I have talked about in the needle that you see, where this is a 15 milliliter tube, where you suck in and blow out the content here. And what is also important to mention here is avoid air bubbles. And I will show you then later how you can avoid the air bubbles in a video, because air bubbles would cause differentiaphorosis. Differentiaphorosis would cause unpredictable homogenization result again. And also avoid aggregates, because every aggregate is bad because it traps organelles, and over-homogenized cells should also be avoided. Now, there are certain cells that are more difficult to get homogenized because they tend to round up, what I said before, or they do not form intracellular contacts. And there is a way out of helping you not to apply too high shear forces, is that you apply swelling procedure by changing the osmolarity of the medium. So basically it's a hypotonic shock. And when you apply a hypotonic shock to the cells, then the cells swell and they become fragile. And with very little shear forces, you can already break them. And this, what is shown here on the bottom, I've said before, with trypsin-treated cells, that you need higher shear forces in order to homogenize them. So at this point, I would suggest that we make a coffee break. Is that okay for you?
Yes.
Yeah, so let's reconvene at 20 past 11 here in the room, and then we will go for a protocol and look at the video which summarizes everything that I've said so far. Think about questions during the break, and during the break and when we take a coffee, I will upload once again this PDF document so that everybody can download the PDF document of the slides I've shown you. Okay, see you later. So, welcome back. Are you back again as well? Give me a sign. Thank you, Nina. Meanwhile, I have loaded once again into the chat room the PDF file. Does now everybody has access to it? I think it was Andrea Lorenz who asked for it. Could you download it meanwhile? Okay, Emma Steiner writes to me just now, sorry, I still can't see it. Okay, let me ask the question a different way. Is anybody in the room who can see it and download it? Lucas wrote, it worked for me. Andre, yes. Nina has worked for me. Leonard for me too. Bettina, yes, it worked. Okay, so maybe Emma, you try again if you can see it or download it. Otherwise, if it is not possible, then send me an email and I will send you a link at the end of the lecture, then that you also can receive it. But basically, I think it is the problem. It has to be solved on your side somewhere, because usually these things work like a charm through the chat that you can click on it and download it. Maybe some of your settings. I'm using Mac system there. With Apple, it's easier, but also with Windows it works usually. Good. Let's proceed, guys. Now with a video. So what you see here at the beginning of the video is a protocol, like one of the protocols that I was supplying to you, or these are the protocols that we use in our student courses. This is the example of experiment 2, a subcellular fractionation, with some introductory words that you can see in here. And then you see the procedure that is described, and you see subtitles coming up. And basically, we will perform that experiment of the homogenization as we have discussed it before. And then we will come to the centrifugation steps step by step. And I will comment it, and you can read the subtitles as well. What I should also say is that this procedure, this video, you find on my YouTube channel. I'm recording the lecture as well, and will put it up onto my YouTube channel so that after the lecture, you can hear it again and you can look at the videos again, or even later when you will be confronted with the situation that you will need information out of that due to your lab work, you can consult my YouTube channel and you find all these lectures also there. Now, just keep in mind, guys, if something is unclear during the video, raise your hand or use the microphone and interrupt me if I don't see it in the chat, or by raising your hand in the interface of the participants. Just take your mic and interrupt me. I will stop the video and then I can add explanations or answer your questions. I will start the video now. So you see the screen for that protocol, things that you have heard already. Here is a list then usually of the materials that you have. Now you see one of these ice buckets. It's already familiar to you. You see that the investigator is going to the incubator, and he takes out now a Petri dish with the cells out of the incubator. This is our student lab, for instance, that you see in the background. You see cells. These are HeLa cells. Passage 26 is written. And he puts it on this ice bucket on this metal plate. You see the instruments available, the scraper, you see the Pasteur pipettes, you see the tubes with the ice-cold buffers. And now what he will do first is he will wash 2 times this cell plate with medium, with fresh medium. And this is in this case a phosphate buffered saline. So he pours off now the medium of the cells. And then you have to work rapidly. The cells don't dry out. You take an electronic pipette aid, you aspirate 10 milliliters or so, and then carefully put it on the side in the tube, in the Petri dish, that you don't wash off the cells from the middle. Just put it on the side on the wall. And then you agitate the plate a bit with the ice-cold PBS, and then you repeat that procedure with 10 milliliters of ice-cold PBS a second time. Like shown here. Blow out the PBS carefully on the wall, agitate it, and pour it off again. Then put back the cells, and now you add 5 milliliters of PBS for the scraping. So you take 5 milliliters of ice-cold PBS, put it in, and this is the sufficient amount of liquid that you need for the scraping. Then you take the scraper. Now you can see this Teflon scraper, and you rotate basically the dish, and the cells will accumulate here. And then you put in the scraper several times that you can take off the cells. You scrape again, and then these little sheets are then becoming visible in here. And this is basically then the solution that contains the scraped off cells. You can see here these white little sheets on the bottom. Carefully transfer it. You see the white little sheet here. For a second, you can see it here again. Transfer that into a 15 milliliter centrifugation tube that you close. And you centrifuge that for 5 minutes at 1,000 rounds per minute, yeah, at 4 degrees Celsius. You go to the centrifuge. The centrifuge is shown here in the back. Oh, sorry, that was stupid for me. So we proceed up to the point where we go to the centrifuge. Then we load the tube with a balance tube in the centrifuge. The centrifuge has been pre-cooled at 4 degrees. You put the settings at 1,000 rounds per minute. You set the time and the temperature at 4 degrees. And when the run is finished, you open the centrifuge, take out the 2 tubes. There should be a pellet then visible in one tube, which are the cells. So we take off carefully. You see the pellet here on the bottom. We take, oh shit. Sorry, that was my mistake. So once again, back to the start. The next working step is to take off carefully the medium without touching the pellet at the bottom, that the pellet of the cells, the white pellet that you can see, stays intact. Usually this is done with a syringe, a 1 milliliter syringe with a blue tip. Carefully aspirate the supernatant here. And this is the pellet here on the bottom. Take off everything. So cells are still intact. Now comes the homogenization buffer, which is a kind of modified PBS buffer. And this contains already the protease inhibitor cocktails, the right salt solutions, and 2 milliliters of that ice cold buffer you put into this tube in a way that the pellet as its own floats up. Yeah, this little pellet floats up in the tube. And this step is now a washing again of the cell pellet in this homogenization buffer. Basically a step that rebuffers our solution with homogenization buffer and protease inhibitors. You generate your balance tube, you go again to the centrifuge, and now you try to generate a tight pellet for the homogenization. You increase the speed to 2,500 at 4 degrees Celsius, and you increase the time to 10 minutes. Meanwhile, you prepare everything, and you prepare the syringe with the homogenization buffer, and you try to avoid air bubbles in the syringe. So you take here 1 milliliter syringe. They're also called tuberculin syringes. These are syringes, you know, also from vaccination procedures, and they are very typical in the lab. You take a needle. In this case, it's a needle with orange color. So this is 22 gauge needle size. And how to fill that syringe is shown in the following. You go into the homogenization buffer and you rinse the syringe once, suck up the buffer and blow it out. And then you turn around, you have the homogenization buffer in there, and you see the air bubbles now accumulating on the top by just agitating this syringe with your fingers, and the air bubbles go to the top, and you blow out these air bubbles carefully in a piece of tissue. And the air bubbles go out, but the remaining part of the needle of the syringe remains filled with ice cold buffer, but there are no air bubbles in there. And now the syringe is prepared for the homogenization step. Now, the tube has been removed from the centrifuge. Again, you take off carefully the homogenization buffer from the top. These were 2 milliliters that you take off, and you have a tight pellet sitting on the bottom of the tube. Now you resuspend this pellet gently in homogenization buffer with 200 microliters per dish, and you use a blue tip for that, because the blue tip has a slightly wider opening than the yellow tips has, because we do not want to break up the cells. We want to resuspend them, that the pellet is entirely solubilized. That you can see here. So you get a turbid solution, a milky solution that reflects that the cells are in solution, and they are still intact. They are not broken, but they are in homogenization buffer with all goodies in there, anti-protease inhibitors. Now you put the syringe in, that it touches the bottom here of the tube, and then you draw up the plunge of the syringe and you suck in the content. And then you blow it carefully out again without generating air bubbles. You can see. And those sucking in, sucking out movements of the syringe, every time you homogenize a number of cells in there. And this step is the step that you should, once you establish a protocol in your lab, you should monitor with a microscope, and then later on you can rely on the number of strokes you use for a particular cell line with a certain passage number, so to say. So this is a 25G needle, by the way, not a 22G needle, I've said before. Good. After homogenization, carefully discard the syringe. And then the next centrifugation step is at 3,000 rounds per minute for 10 minutes. And this step pellets out the nuclei and cell debris. So aggregates of unbroken cells which are still remaining in there, or larger aggregates that you would not like to have in the procedure. And then you have this post-nuclear supernatant. On the bottom, you have the pellet with the nuclei and the unbroken cells. Then you take out an aliquot of this post-nuclear supernatant for analysis or for loading it on a gel. When you compare the post-nuclear supernatant, maybe then biochemically by gel electrophoresis and Western blotting afterwards with the subcellular fractions that you would like to purify out of that homogenate. And you transfer that in an extra tube. Now here, you can take out 100 microliters, for instance, and you would transfer that 100 microliters in an ultracentrifugation tube, for example, to centrifuge that at 100,000 G. So what would that first very simple fractionation achieve? With 100,000 G centrifugation, you would accumulate at the bottom of the tube in this ultracentrifuge rotor all cellular organelles, all cellular membranes, and this is the so-called P100 pellet then. And everything that is above the pellet in the supernatant would be cytoplasmic content, cytoplasm with protein machineries that would be large, but not that large that they would pellet at 100,000 G. We will talk a bit about ultracentrifugation during the course of this next 20 minutes or so. You close the ultracentrifuge and you turn on 100,000 G in a pre-cooled ultracentrifuge, and then the vacuum is built up and the centrifuge starts for 1 hour at 4 degrees Celsius. So we have now, in a very simple step after the homogenization, prepared the total cytosol fraction of the cells and the total membrane fraction. This allows you already to answer simple questions your supervisor might impose on you because he is interested in your thesis work. If a receptor or a protein shuttles from the membrane fraction into the cytoplasmic fraction about a certain cellular stimulus, for example, a growth factor, a differentiation factor that you add on the cells, and you would see before or after addition the localization of this receptor or protein changes because it shuttles between the cytoplasmic compartment, the S100 fraction, or the membrane compartment, the P100 fraction. As simple as that. Now, there are other methodologies where you can break up cells, and we had mentioned one of the problems already. These are round cells or cells that are difficult to break up. Or what you could also imagine could become a problem is large quantities of cells. So imagine you are hunting very low abundant proteins. And there is a natural limitation of the amount that you can analyze. And your supervisor is very unsatisfied with your work and says, come on with this syringe approach, with these small amounts, milliliters or so. We will not get any further. We need higher amounts of the protein. The protein is very low abundant, but we need higher amounts for subsequent biochemical analysis. So you start growing cells to large quantities. And these large quantities can be achieved in cell factories, for instance. So these are cultures that grow in bottles that roll all the time. And these cell factories, they can be placed into an incubator, and you have these roller bottles, and with these roller bottles, you rotate the bottles all the time. So when you put in HeLa cells, for instance, or other cells, the cells adapt after some time to the condition that they cannot attach to the plastic surface. And after the adaptation, they start dividing in the medium. And the medium is rotating, the medium is humidified in the incubator, it has the right oxygen concentration, the right CO2 concentrations, and the bottles are rotating all the time, and you can, within rather short times, generate large amounts of cells. And this medium where the cells are in there, you can harvest. And of course, there is now a limit with this approach with syringes, because you are generating hundreds of milliliters of cell suspension with a high density of cells. And if you would work that off with a 1 milliliter or 2 or 5 milliliter syringe, there would be, you know, an end of joy for this experiment reached rather soon, and the level of frustration would be rather high. So one way out of that is to use nitrogen decompression or nitrogen cavitation. And what is that? You have steel vessels that can be closed firmly, and which have an inlet valve and an outlet valve and a barometer for measuring the pressure. And what you can do in such an open steam vessel, you can fill in your cell suspension. So this is a steel vessel here for about 200, 150 milliliters. And these steel vessels can be obtained in different sizes, up to liters. They can be purchased. They are not very expensive. What you need is access to nitrogen. So you need a nitrogen bomb, a flask of nitrogen in the lab. Then you can perform that on your bench. You would fill your cell solution in there. You would close it, and then you would let the nitrogen come in. So what happens then? You increase the pressure in this vessel, and you let large quantities of nitrogen flow into this vessel. And what happens is that the nitrogen under this high pressure makes a phase transition from the gas phase into the liquid phase and gets into the liquid phase. And that means the nitrogen is now in the liquid phase, and you have this cell solution where the nitrogen is in the liquid phase, and it also is in the cells. When you release this pressure, what happens is identical to what happens when you have a diving accident. Through this sudden release of the pressure, the nitrogen undergoes a phase transition again from the liquid phase back into the gas phase, and little gas phase bubbles are generated, and those bubbles and this ray transition from the liquid phase into the gas phase disrupts the cell all of a sudden like an explosion. And this is what happens to the divers when the divers go deep into the sea. And they dive up too rapidly. This transition from the liquid phase to the gas phase happens too fast, and then the microvessels are bursting, the capillaries are bursting, and that's the reason why in these diving accidents you destroy the organs by the capillary pressure that is increased to destroy the capillaries, and that's the reason why these divers start bleeding and why they have to be put into pressure chambers back again for a certain amount of time in order to prevent this happen or to slow it down and to prevent the tissue damage. So here you see the procedure on the bench. You pour this 50 milliliter solution into that steel vessel, then you close it, you hook it onto the nitrogen supply for this tube, and then you let the nitrogen flow in. You have the pressure barometer, you let it go up to 200 bar or something like that. And then you open this valve, you close the valve of the nitrogen that was flowing in, and you open the exit valve here on the bottom. And then an explosion is taking place within this vessel, and the solution is pressed out, and all cells are homogenized through this pressure. And this sounds rather brutal to you, but it has many advantages. Surprisingly, on first sight, but when you think about it more deeply, it's not so surprising. It's a very gentle method because you have no chemical stress on the cells and you have no physical stress. That means you do not generate heat damage due to friction for pressing it for a pistol or for pressing it to a French press or for a syringe. Therefore, no oxidation of the sample takes place. And you can use any suspending medium right away. That means you can treat your cells with an inhibitor, a chemical inhibitor, or with anything else, with a growth factor, take them out of the incubator, pour the solution with the cells, with the medium directly into the chamber, and you do the fractionation directly into the medium, and then you can purify everything out of that. The huge advantage is that every cell is exposed to that only once, and the product is therefore rather uniform, and it's very easy to apply. Do you have any questions to this nitrogen cavitation so far? Or is that clear? Good. With this, we close the chapter of the homogenization. And we have learned that homogenization is the crucial part of your sample preparation. We have heard about all the precautions that you have to take, that you have to preserve your sample, that you have to keep it in ice, that you have to add protease inhibitor, that you have to apply gentle conditions, not to disrupt other organelles, to keep them as intact as possible, but to disrupt the plasma membrane and release the content into solution. We have seen a video how you apply that, and we come now to this last part of the video where you start separating these organelles and where you're using gradient centrifugation for that. Now, the principle of density gradients is that during the centrifugations, organelles will sediment or float until they reach their isopycnic position within the density gradient. That means the isopycnic position is where the organelle, due to its physical properties that are defined by the protein content, the lipid content, the charges, and the shape of the organelle does not move anymore because the physical environment has the same density and the same specific weight as the organelle has at that moment. The problem I have pointed out at the beginning of the lecture is has already been mentioned. It is some compartments share, unfortunately, similar physical properties. And therefore, we are using different media for this separation, and you can use sucrose, for example, Ficol, Bercol. These are commercially available density media, Nicodens, metrisamide. The easiest is sucrose solutions that you can prepare with different densities. and that you can overlay over each other, or you can make step gradients with 1 step, 2 step, 3 steps, or you can make a continuous density gradient. And there are gradient makers that you can use, which are basically machines that rotate tubes with a sucrose solution under a different angle. And depending on how you put that angle and the speed of the rotation, you can generate gradients from 10 to 40%, from 5 to 30%. That goes all automatically, and these gradient makers are everywhere where molecular biology is performed, or where, for instance, gradient centrifugation by cell biologists are performed. This is all automatic, and you can adjust the instrument. For sure, you have made gradients already yourself during practical courses in molecular biology or biochemistry, that you overlay sucrose solutions with different concentrations. You start with the highest density on the bottom, with 30%, so to say, and then you overlay a 10% or 5% sucrose solution carefully with the pipette, that the interfaces don't mix with each other. But that is the principle. Now, with the gradients itself, you have different possibility. One possibility is a fixed angle centrifugation. So basically, you have a rotor where the tubes are inserted in a position with a fixed angle, and you can see here from the distribution of the blue color that the organelles would separate asymmetrically here, not in a straight line, which is perpendicular to the tube wall, but in a diagonal line. This is important when you take them off later. In this chamber, the motor and the spindle is on the bottom. You put the rotor on top of that. You have a refrigeration system, and you have a vacuum system. Certain speeds in a centrifuge can only reach when the rotor is rotating in a vacuum environment. And there we are talking about speeds of about 100,000 G and above, for 30,000, 60,000, 100,000 G. And that has to be generated in a largely friction-free environment. That means in a vacuum. And on top of that, you need to cool the system. And the second possibility are swinging arm centrifugation rotors. So basically, you have a second type of rotor, which has the inlets for where you put the tubes in a flexible way that during the centrifugation, they would swing out due to the centrifugation forces. And then, of course, you would generate sedimentation profiles which are perpendicular to the tube wall, and the centrifugal force is applied in this direction, and you would pellet or float then the organelles. The heavy stuff would pellet on the bottom, the light stuff would float up. And when you look at the cell homogenate here before the centrifugation, you see the equal distribution of the organelles in there. And after the centrifugation, like it was the case in normal buffer, not in a density gradient, like we saw before, the 100,000 G pellet, you would have the large and more dense components like all membranes, all organelles, all large protein machineries like ribosomes pelleting to the bottom, whereas in the supernatant, we have smaller and less dense components and the cytoplasmic proteins. Now, some words about the care and the maintenance of centrifuges. This is something that I would like to teach you today, which is important, and I would like to mention it. So centrifuges are expensive pieces of equipment. Their costs are up to the costs of a Porsche or a Mercedes of the higher segment of cars. So they are very expensive, and they are sensitive, and you need to take care of them. So what is a problem with centrifuges and with the mobile parts of the centrifuges, which are the rotors? Well, they are all made of heavy metal pieces and parts, and therefore corrosion and degradation due to the quite aggressive biological buffer systems you are using will damage rotors and can contaminate the interior or exterior of the centrifuge due to spillage, and this may affect the lifetime of the equipment. Another very important point where my laser pointer is now resting here on the left side is the proper balancing of the centrifuge tubes. And this is important for safety, but when the centrifuge tubes are not properly balanced, it might cause vibration-induced artifacts and damages of the rotor itself. And the spindle where the rotor is resting on. You should not forget that these centrifuges spin with 100,000 G or more. And G is the centrifugation force in the Earth acceleration measured. So this is a huge force that you apply. And I remember a case where a centrifuge has exploded. Nowadays, centrifuges have built-in safety measures that when a bit of vibration is sensed in the centrifuge, it stops immediately. But years back, for instance, when I was a young postdoc at the European Molecular Biology Laboratory at the EMBL in Heidelberg, a centrifuge exploded in a laboratory underneath us, and the rotor came out like a rocket from the centrifuge and drilled into the ceiling of the room. And we could hear the explosion in the entire building. So this can be extremely dangerous instruments. Nowadays, they have safety measures that those things should not happen anymore, but you destroy the centrifuge. And we are talking here about instruments that can cost about 100,000 euro. Therefore, proper handling and care and regular maintenance of both the centrifuge and the rotor is a very important part of keeping this biochemical method available in the laboratory for you. And keep in mind what I have told you. Learn how to use these machines. So when you come to a lab and you do your first centrifugation round, ask an experienced researcher in the lab, a postdoc or PhD student, or the technician in the lab to show you how to deal with the instrument. How long does it take until the vacuum is built up, and why you should keep the rotors cool at 4 degrees in the cold room, what measures you should take to clean the rotors afterwards, which liquids are you allowed to use for cleaning the rotors, which are not too aggressive and cause corrosion or degradation of the material, and many other things that you should take into consideration. Take your time, learn how to use the instruments, and then the instrument will serve you a perfect task and will help you to solve your scientific questions, but you can destroy it when you don't take these measures into consideration. The rotors, as I said, should be kept at 4 degrees. First, they should be dried, and you should take out all liquid that has accumulated, like condensed water, and put them on a dry place. Before you use them, you should pre-cool them, and you should clean them regularly. It is important that the laboratory invests into regular maintenance and service contracts for rotors and centrifuge. Engineers come frequently to the labs. They test the rotors, they test the centrifuges, because rotors, when they reach a certain age, they have a bit of corrosion, and when they corrode, then the weight is not equally distributed anymore. And these little weight imbalances cause tremendous problems for the machines when you think about the acceleration of 100,000 G, for example. You need an accurate record keeping. So that means usually when these centrifuges are in common use areas of institutes, you find a calendar booking system there where you sign in with your name, with your room number, and from when to when you have to use the centrifuge. This is very useful because when you come to the centrifuge and you want to do your experiment and the rotor is in bad shape or has not been cleaned, you can look at the calendar, at the booking system, and know exactly it's the colleague next door to you who has left the rotor in such a bad condition, and you can come back and chase him and tell him he should clean his mess before he hands over the instrument back to you. So these are words of precaution. Be careful when you use centrifuges. What you have to avoid is mechanical stress, and that means that the loads have to be evenly balanced before a run. And that usually you do with a balance and not by eye. And don't forget, when you load gradients, the balance tube on the other side of the rotor that you need to counterbalance your weight cannot just be filled with water. It has to be filled with an entire gradient, with the identical gradient. And you have to balance that drop-wise, the weight, by putting the buckets with the tubes on a balance in a glass or whatever, in a beaker, and measure it that you have really achieved with droplets of homogenization buffer or sucrose buffer the same weight before you put them in the centrifuge. Be sure that you know what is the maximal speed you can go with a centrifuge and with the tubes that are supplied, because what happens is when you take the wrong tubes that are not designed to stand at centrifugation speed of 100,000 G, they will collapse and all the liquid will distribute then in the centrifuge. The rotors have to be taken care of corrosion. Many motors are made of titanium and aluminum. Aluminium is not corrosion resistant, whereas titanium is. And when corrosion occurs, this weakens the metal surface, and the stress that is applied by 100,000 G centrifugations is tremendous. So be careful. And if you see that the rotor looks damaged from outside, don't use it, and consult immediately with more experienced researchers and technicians in the lab. Now for the centrifugation, there is a possibility that you can put several centrifugation steps after each other in a row. And that looks like this. You have, for instance, a cell homogenate in a tube with large organelles, small organelles, or even intact cells that are still not completely broken up, or cell debris. And you apply a low-speed centrifugation first, like we have said before. You remove the whole cells, the nuclei, and the cytoskeleton aggregates. This is what I called before the post-nuclear supernatant, because here the nuclei would be, and here is what the post-nuclear supernatant. This supernatant, after medium speed, after this low-speed centrifugation, you can apply to a medium-speed centrifugation, and you can spin out mitochondria, lysosomes, bioxysomes, everything that is a bit larger. And then you get a second supernatant that you can apply to a high-speed centrifugation, where you spin out microsomes. So these are vesicles of the ER and small vesicles. And then you do a very high-speed centrifugation with the remaining supernatant, and you can spin out single ribosomes, virus particles, or large protein machineries and large protein complexes. So simply by putting different centrifugation speeds one after each other, you can achieve a remarkable degree of separation already. You can use also density gradients, like shown here from one of the protocols, where you load your post-nuclear supernatant, for instance, on the bottom of the tube in 40% sucrose, and you overlay that with 35% sucrose, 25% sucrose, and finally homogenization buffer, which has only a very few percent of sucrose. That means you have now different layers of density, and when you put such a tube in a swing-out rotor and centrifuge it, organelles will accumulate at these interfaces of these different sucrose solutions. So Golgi and EDR membranes would accumulate in the first interface, early endosomes, carrier vesicles at the second interface, late endosomes and lysosomes here rather on the top of the gradient. And these different sucrose solutions, of course, when you put them on top of each other, they should also be kept on ice. And when you overlay them, it's advisable that you take a water-insoluble pen that you use for labeling your lab tubes, which is water-insoluble, where you mark these interfaces where, for instance, 35% of sucrose is overlaid with 25%, or where 25% is overlaid with 10%, because afterwards, when you take the tube out after the centrifugation, you know exactly the position of the interface where these membranes should accumulate. Usually, when you have sufficient amounts, and in the lower areas of the gradient, you will see a milky interface. And the milky interface is generated by the membrane that's accumulated there. It's visible with eye. However, when you have smaller centrifugation tube and smaller amounts, it is helpful that you mark your tube from the side. And this shows you such a swing out rotor, a bucket. These are these buckets. Every lid is numbered and every bucket is numbered because the netto weight of bucket number 1, so to say, plus lid number 1 should equalize bucket number 4, that is on the opposite position than in the swing out rotor, like shown here, 1 and 4 here. And with the lid number 4 and the bucket number 4. Here you put your tubes in, and this entire thing is then balanced on the balance area that has the same weight. After the centrifugation, when you remove the lid from the bucket and you take out the tubes, you see these milky interfaces accumulating there where you have made your little marks with the water-insoluble pen. And these are the membranes that you can then take off. These are the purified organelles that you can take off and analyze further. And now, before we make another little break, I will show you a video that summarizes this procedure in the subcellular fractionation that we have just heard now, and just look to that now.
In subcellular fractionation, cells are disrupted and their components are isolated into relatively homogeneous fractions. The first step in subcellular fractionation is breaking the plasma membrane. A high-speed blender can break the plasma membrane and endoplasmic reticulum into small fragments, while leaving nuclei, mitochondria, and other organelles intact. The suspension of broken cells is called a homogenate, or a lysate, and is now separated into like components by a series of centrifugations. An ultracentrifuge can rotate samples at very high speeds, over 100,000 RPM, to produce forces up to 500,000 times greater than gravity. However, the first spin is usually set at a low rotation speed, producing a centrifugal force of about 800 times the force of gravity for 10 minutes. During this spin, the largest and densest materials sediment first, forming a pellet at the bottom of the tube. This pellet will contain unbroken cells and nuclei. Thus, an enriched fraction of nuclei can be recovered from the pellet, while the other cell components are suspended in the remaining solution, called the supernatant. The supernatant is now centrifuged at a higher speed, producing a force of 15,000 times the force of gravity for 10 minutes to sediment mitochondria, lysosomes, and peroxisomes. The supernatant contains smaller and less dense components. Recentrifugation of the supernatant at an even higher speed and duration sediments fragments of the plasma membrane and the endoplasmic reticulum. A fourth centrifugation at a still higher speed sediments ribosomes, leaving only the soluble portion of the cytoplasm, the cytosol, in the supernatant. This series of spins at higher and higher speeds and durations is called differential centrifugation. In differential centrifugation, subcellular components are separated by their size and density, with larger and denser components sedimenting faster than smaller and less dense components. The fractions obtained from differential centrifugation correspond to enriched, but still not pure organelle preparations. For instance, some smaller particles will be found in all pellets, considering some of these particles are already present at the bottom of the tube in the original homogenate. A greater degree of purification can be achieved by resuspending the pellets in solution and then performing density gradient centrifugation, in which the sample is made to sediment through a gradient of a dense substance, such as sucrose. At the top of the gradient, the sucrose concentration is lower than at the bottom of the gradient. There are two main types of density gradient centrifugation: velocity centrifugation and equilibrium centrifugation. In velocity centrifugation, the sample is added carefully to the top of the tube in a narrow zone. The sucrose gradient below the sample serves to prevent the convective mixing of the solutions and keeps the particles in tight zones during centrifugation. Particles of different sizes sediment through the gradient at different rates, moving as discrete bands. In this technique, the density of sucrose at the bottom of the tube is less than the density of the organelles, so the centrifugation is stopped before the particles can form a pellet at the bottom of the tube. Following centrifugation, the different zones in the tube can be isolated in separate collection tubes. A hole formed in the bottom of the tube allows the collection of the different fractions. The fast sedimenting particles emerge first. The slow sedimenting particles emerge later. The other type of density gradient centrifugation is equilibrium centrifugation. In this technique, the concentrations of solutes in the gradient are very high, such that the density of the gradient at the highest point is higher than the density of the particles of interest. Organelles with higher densities are defined as having greater mass-to-volume ratios. During centrifugation, the particles move to the location at which their own density matches the density of the surrounding solution, and this is where they remain. Although larger particles may travel faster to their equilibrium locations, the spin is kept going long enough for all the particles to reach equilibrium. Thus, the final state of the tube depends only on the buoyant density of the particles. Equilibrium centrifugations are useful in separating different types of membranes from one another and are sufficiently sensitive to separate macromolecules that are labeled with different isotopes. A classic example is the analysis of DNA replication by separating DNA molecules containing heavy and light isotopes of nitrogen, using equilibrium centrifugation in cesium chloride gradients. The light and heavy DNA come to equilibrium at different locations along the tube. A hybrid DNA with one light and one heavy strand bands in the middle.
So, my suggestion is now that we make a short break and refresh a bit before we come to the next chapter, and we will reconvene 5 minutes before 12:30. So in 12 minutes, something like that.
Okay.
Thank you. So, welcome back, my friends, to the show that never ends. This is an original citation from a very famous rock band where they opened a concert in the early 80s, Emerson, Lake and Palmer. Maybe a few of you might know them. History of rock. So welcome back to our lecture. A few more words about quality control for the purification. The drawing here shows you a scheme of endocytosis. And you see here receptors at the plasma membrane, tyrosine kinase receptors, for example, like the epidermal growth factor receptor, EGF receptor, which plays a major role, for example, in the maintenance, establishment of all epithelia in our body, but also during the course of cancer development, for instance, like in colon cancer or in breast cancer. Now, this receptor, once it is engaged with its ligand on the plasma membrane, is phosphorylated and activated and transported through endocytosis, through cluttering-coated bits, into transport vesicles which fuse with an early endocytic compartment where the receptor is still active. And then later they are transported by vesicles, multiple vesicular bodies, late endosomes towards the lysosome, where finally the receptor is degraded and the signal is shut off. Now, when you're interested, for instance, in purifying those compartments and looked at the activity state of the receptor during its traveling, or the composition of these compartments, how they might change during signal transduction and engagement with growth factors when cells engage in a different transcriptional program, then finally you apply subcellular fractionation to separate early endosomes from late endosomes, for example. And with the different density gradient steps and purifications I have shown you, you are able to separate those organelles, the early endosomes, which have a certain density on these gradients, from late endosomes and multivesicular components, which have a bit of a less density and migrate at a different position in the gradient, whereas those organelles you find more in the middle of the gradient, those organelles you find on the top of the gradient because they are lighter due to their different lipid composition. Now, you can visualize that by a variety of different technologies. And one is shown here for you, and I'm showing this technology here for didactic reasons. It's because it visualizes the purification very nicely. So this is a two-dimensional gel electrophoresis. Now, just to back you up a bit on that technology, that means you separate you separate proteins in two dimensions. The normal gel electrophoresis principle is known for you. That is, you separate on STS pages according to the molecular weight from the top to the bottom of this gel. So you have high molecular weight proteins up here, low molecular weight proteins down here. In two-dimensional gel electrophoresis, you separate them in two dimensions. In the first dimension, you separate them in tube gels according to isoelectric point. So acidic proteins are on the left, basic proteins are on the right. And then you put that tube gel on top of a normal STS page and you separate in the second dimension the molecular weight. So you get a two-dimensional spreading where each spot has a unique position because each protein has a unique isoelectric point and molecular weight that it might share with other proteins, but then you have a second separation that the isoelectric point is different. What you can do with that technology is you can label your fractions with fluorescent dyes. So, and then you can mix different fractions in one gel, and you can visualize the origin of the fraction that you are looking at with the different color that you have labeled your fraction beforehand. So in this experiment, what we did is we labeled the post-nuclear supernatant with a psi dye, which is called psi-2, which is in blue. The early endosomes after the purification, we labeled with a different psi dye, with psi-3, and that's green. And with psi-5 in red, we labeled the late endosomes. Then we mixed those fractions and separated them in 2 dimensions in these gels. And now you see blue proteins, which are only in the post-nuclear supernatant, which you leave behind when you purify early endosomes, because then they become green. So these are proteins that are only seen in early endosomes. Or in red, then they are late proteins. When they are in both fractions or in all 3 fractions, then they represent the color, which is the overlap. For instance, proteins which are common in early and late endosomes appear as red, because green and red, when you overlay it, becomes yellow. Magenta is the fraction where post-nuclear supernatant and all 2 of them, and 1 of them is contained. And in white, they are when they are in all 3 fractions. And this is what you see with your visual eye already. When you scan that gel with fluorescence scanners, you see approximately 7,000 to 8,000 protein spots there, which comes already rather close to the proteome which such an organelle can contain and which you can resolve. Now with robotics, you can cut out these single spots, and you can analyze them in a mass spectrometer, and you can identify them, but you can also quantify them, and you can see how much enrichment you can get. And when you quantify that by fluorescence intensity, you find up to 120-fold of enrichment in the late endosomal fractions, but only 25-fold enrichment in the early endosomal fractions. And you find, of course, different numbers of proteins that are present in all kinds of fractions distributed more or less equally. And this picture already shows you several things. You can enrich certain fractions very nicely, but you have also a large amount of proteins that show up in different fractions. They are either contaminants that you carry along coming from other organelles, which are rather abundant in the cell, like the endoplasmic reticulum, or those proteins that are by their nature present in different organelles. And such a procedure that you can then fine-tune biochemically and learn more and more with the simple technologies just by putting them after each other is shown here. Where you start with a cell culture, the collection of the cells, the homogenization, the total cell lysate as you have as a first fraction, the post-nuclear supernatant with the nuclei and the post-nuclear supernatant here. And then you take this supernatant, you further fractionate it. Either what I have shown you in the video, you make 100,000 G pellet and you have the total membranes here and the cytosolic fraction, or you use continuous or discontinuous gradients to further separate late endosomes from early endosomes, or you purify common endosomes, the total endosomal fractions. Biochemically, you can further dissect these membranes. For instance, you would like to know the nature of these membrane proteins that you recover in the total membranes. Are they integral membrane proteins spanning the membrane several times and being tightly associated as integral membrane proteins, transmembrane proteins, or are they peripherally attached membrane proteins by using, for example, sodium carbonate extraction at high pH, because when you extract with sodium carbonate at high pH, all proteins after such a centrifugation at 100,000 G that show up in the supernatant are by definition peripheral membrane proteins, which are associated with the membrane by protein-protein interaction or protein-lipid interaction, but are not integral membrane proteins because they would only remain in the pellet. You can use marker analysis by Western blotting. You can use enzymes. There are enzyme kits available for all kinds of enzymes that are present in peroxisome, lysosome, endosomes, that you can use to benchmark your gradients where you are and which fractions you have enriched. And you can analyze those fractions by gel electrophoresis, chromatography coupled to mass spec. The sodium carbonate extraction that is shown here as point 1, where you can dissect the nature of the membrane proteins, is visualized here by a two-dimensional gel electrophoresis, the technology that I introduced just before. Here you see a 100,000 G pellet showing you all integral membrane proteins, the one where I've put circles around, we have confirmed by mass spectrometry, we know how many transmembrane domains they contain. And here on the right-hand side, you see 100,000 G supernatant of the same total membrane fraction that was derived after this ultracentrifugation step that I have shown here. And these are all the peripheral membrane proteins. And when you put these two images on top of each other, then you get an estimate how many membrane proteins are present in such an endosomal fraction. And these are many thousands of them. Again, coming back to the initial question that we have asked before, how complex is the proteome and how can we dissect it? And now we are getting a closer step to that because we can visualize these proteins, we can see them, we can identify them, and we can enrich them. Now, for that, we need, of course, markers that we introduce to follow our fractionation procedure. When we remain for the didactic example of the endosomal fractionation and purification, we have drawn here the endocytic pathway and on the left side, the biosynthetic exocytic pathway. Now, these pathways communicate, of course, with each other. We have pre-existing compartments in the periphery of the cells, which are early endosomes, and we have pre-existing late endocytic compartments in the perinuclear region of the cells, like the lysosome or the late endosome. And these two compartments are linked by a vesicular transport steps that utilizes the cytoskeleton with motor proteins. And this is the multivesicular body or endosomal carrier vesicle pathway, which utilizes microtubuli and microtubuli motor proteins, tiny in motors, to migrate towards the perinuclear region or to migrate outwards of the cell in the secretory pathway by using kinesins, for example, so differently directed motor proteins. The advantage of this endocytic system is that by nature of endocytosis, you can access it from outside. So you can bring in fluid phase markers. These fluid phase markers can be enzymes like horseradish peroxidase, which is an enzyme, when you put a chemical on it, diaminobencidine, it forms a brown aggregate and you can visualize it. You can visualize it in an electron microscope and you can visualize it biochemically to its brown color in a photometric approach that you can, for example, measure photometrically the density of it after gradient centrifugation into different fractions, and you can calculate the amount that you have internalized. You can also use fluorescent dyes, as we will see later in this course, to fill these compartments with fluorescently labeled ligands, where we decorate receptors and internalize fluorescently labeled ligands like EGF with the EGF receptor into this early endocytic compartment and later in the late endocytic compartment, and we can follow those markers. On top of that, we have certain proteins which are specifically attached to these compartments. The early endosome, for instance, has two parts. One is the recycling part, where proteins recycle back to the plasma membrane and are reutilized here from this tubular part of the endosome. And the more globular part of the endosome contains GTPases like RAP5, the transferrin receptor. Other parts contain also the transferrin receptor when they recycle back, but other GTPases which are specific for the recycling like RAP11, RAP4, or early endosomal antigen number 1, EA1. And the same thing is true for the late endocytic compartment and the multivesicular bodies. It's true for the Golgi, it's true for the ER. Here we have, for instance, RAB7 or an enzyme that we can easily read out with kit biology, where we can buy kits from Sigma, Aldrich, or any other supplier, for example, beta-hexosaminidase. So that means we have tools at hand that we can follow precisely what we purify on the gradients and which organelles we have at the end of the day in our hand. Now, the advantage here is also that we can use this internalization and we can manipulate the internalization, because we know that the transport from the early endosome to the late endosome requires an intact microtubule network. And we can disrupt these microtubules, and we can block at this position and do not allow these vesicles to fuse with this position, for instance. And how is that done? So we know to reach early endosomes when we internalize a marker, it takes a few minutes only, 5 minutes, so to say. Then we have reached the early endosome. Now, in order to enrich these carrier vesicles that in this graphic link the early endosome to the late endosome, we need to enrich these vesicles, because otherwise they would bud off here, be transported very rapidly to the late endosome and fuse with the late endosome. And when we want to analyze those vesicles only, we have to disrupt the way from here to here by disrupting the microtubules, and that we do with a drug which is called nocadazole, which depolymerizes microtubules. So in such an experiment, what you do is that you internalize for 10 minutes your ligand, your marker that you want to follow, and you chase for another 30 minutes and wait another 30 minutes before you fractionate the cells upon disruption of the microtubules, for instance, by depolymerizing them. The nice thing is that this step is reversible, so you can wash out this drug, this nocadazole again, and the microtubules would polymerize in real time again. But we are applying that in order to disrupt the microtubules and to accumulate these carrier vesicles. So I would have now one dish where the cells are internalized for 5 minutes, then I put them on ice, and I stop every transport. I disrupt the cells and purify the early endosomes where the ligand, for example, the EGF, the labeled EGF or the horseradish peroxidase was internalized. Then I have a parallel dish where I want to purify the carrier vesicles from the same cells. Here I internalize for 10 minutes. And then I disrupt the microtubules on ice and I accumulate those vesicles, and then I put the cells on ice, and these vesicles have accumulated now, but due to the fact that the microtubules have depolymerized, they cannot fuse with the acceptor compartment. So I accumulate these vesicles and can purify them out. Or I internalize for 10 minutes, is 30 minutes chase, without addition of this microtubule depolymerizing drug, and then everything will be in the late endosome, and then I can follow on the gradient the late endosomes and can purify them. And that means this marker analysis provides us with a quality control check. Because in all fractionation experiments, a kind of balance sheet has to be established. For the distribution of the protein markers, like the ones that we internalize, like horseradish peroxidase, or the ones that we read out by Western blotting and want to quantify. So this provides us with an appropriate means to judge the fractionation steps, the homogenization steps, how efficient are they, how reproducible are they, what kind of enrichment can we achieve, and helps us to compare preparations from different days of the experiment. On top of that, it helps us to introduce quality measures that those technologies that we introduce are reproducible in other laboratories. And this is finally very important for you when you publish your results and want to have your results available for the scientific community to repeat them and to use them. So you need this kind of quality controls. And this is like such a balance sheet would look like. This is just an example. I guide you briefly through that you see how one adjusts an Excel sheet or a matrix for that. On the left side, we have the fractions. They are numbered. Yeah, and the fractions we collect, so these are 15 or 20 fractions we collect, and the starting fraction is the post-nuclear supernatant, and the light fractions are on top, the heavy fractions are on bottom of the gradient, and we collect the gradient from top. Therefore, the low numbers are on the top of the gradient, the high numbers are more on the bottom of the gradient. So we have late endosomes, early endosomes, lysosomes, here on top, the late endosomes and lysosomes, the early endosomes, and the other membranes. In the middle up to the bottom, and the very heavy fraction is the post-nuclear supernatant. You record what the fraction size is in microliters, the volume. You measure the protein content. That gives you a protein concentration that you recover, because the protein amount per volume gives you the concentration of the proteins. You know which amounts of enzyme, for example, horseradish peroxidase, you have introduced and recovered in the fraction. From that, you can calculate the specific activity that you have in there, and from that, the enrichment. And from the enrichment and the total HRB that you have introduced, then you can calculate then the yield, and you see the yields are rather different. And from that, you see how much you can enrich your fraction and how reproducible is that and how are the yields. But this is just an example, and that can be adapted and adjusted to your needs. What I'm trying to say is document carefully how much you enrich and how you quantify your Western blots, how you normalize that. You need to get a feeling how reproducible is your technology. And that is shown here, for example. Such a gradient where we separate late endosomes from early endosomes on a continuous gradient from 10 to 40%, 10 on the top, 40% at the bottom. The organelle markers you read out here with Western blot, that are markers that are found on both endosomes, late and early endosomes, or are markers that you found either on early endosomes or on late endosomes. Let's look here at the bottom with the Western blot P14. This is a protein that you mainly find on late endosomes and lysosomes, and it should not be on early endosomes. So far, our separation is good. RAB7, as I have shown you here before, is a GTPase that is found on late endosomes. RAB5, RAB4 transferrin receptors is found on early endosomes. So let's look at our gradient and on the Western blots, and there we see RAB5 mainly on early endosomes, but also a bit on late endosomes. Transferrin receptor only on early endosomes. LAMP1, lysosomal associated membrane protein number 1, mainly on late endosomes. So that gives you a good feeling that you can enrich to a certain extent, and that this is already rather useful for you. When we look here at the distributions, for example, of the HRB internalization and where the HRB is recovered, is it on the plasma membrane sticking? Is it internalized in early endosomes and then late endosomes? We can see here the peak where the late endosomes are. So these are these fractions here, 6, 7, 8, 9, then this peak here. We see a nice internalization, but we see there's a problematic area in the gradient where we have different endosomal fractions, plasma membrane, all accumulating in that area. That means here we have a rather low resolution, here we have a good resolution, showing you that you need this kind of balance sheets in order to be sure how much you can enrich. When we look, for example, at purified nuclei, these kind of contaminations that we carry along become obvious. Here is a purified nucleus after the post-nuclear supernatant. This is cell debris that we recover here. And when we zoom in with the scanning electron microscope on this nucleus, we see a lot of membranes sticking on this nucleus. So now what are these membranes? So these are membranes of the endoplasmic reticulum. And you know, there is no surprise why the endoplasmic reticulum would distribute with the nuclei, because the outer nuclear membrane is in continuum with the endoplasmic reticulum. But it shows you also that the endoplasmic reticulum is a major organelle in the cell, and it will distribute in many parts of the gradient, not only on the bottom of the gradient. So all these technologies that we put behind of each other can achieve a certain degree of purification, as you can see here, but they are still not perfect, but they are already absolutely useful and absolutely required to ask certain biological questions. But this brings us to the next chapter, and the chapter is how we can improve either the gradients or can we introduce different methodologies based on different principles in order to achieve for certain questions better separations. And one logic extension of the density gradients are so-called density shift gradients. So what are that? In the density shift gradients, for example, when we look at endosomes, we can load endosomes from outside with nanoparticles. For instance, colloidal gold that is bound to a ligand like transferrin or EGF that is hooked to colloidal gold particles, which can have a nanometer size, let's say from 10 to 50, 60 nanometers. And they can be loaded into endosomes by endocytosis. They bind to their respective receptors, like one scientist, Colin Hopkins, has shown it for transferrin. And then those fractions containing these colloidal gold particles change their behavior in the density gradients because they are a bit heavier and migrate therefore differently, and thereby you can shift these fractions a bit away from the problematic area in the middle of the gradient. Also endosomes where we have loaded these enzymes, horseradish peroxidase, bound to a ligand because you can label any ligand with this enzyme. When we add diaminobencidine to cells, for instance, that have internalized this ligand, or to endosomes that have been purified in the post-nuclear supernatant, for example, and perform this reaction, then an aggregate is cross-linked and proteins are cross-linked in the lumen of the endosome. And by that, the physical behavior of the endosome during the gradient centrifugation changes in a way that they float to a different position in the gradient. And by this cross-linking can be then collected. So these are simple tricks like density shifts. Now here, I introduce you to something which is more sophisticated and which is called free flow electrophoresis. Now, free flow electrophoresis is something like a purification through a medium based on an electrophoretic principle that you apply a current and separate protein complexes, organelles according to their charges. It's a very powerful preparative separation tool for protein enrichment, and it's especially suited also for very complex protein mixtures. The principal idea is that according to the load of the lipids and the proteins and amino acids, every organelle has a certain netto charge. And according to that netto charge, it can be deflected in an electric field. So isolation of subcellular compartments or pre-fractionation of complex protein mixtures based on these charges uses pH gradients that can be applied. And for example, also lysosomes and endosomes can be separated. And the sample submission is very easy because it can be supplied in buffers with a low salt concentration. Like for every electrophoretic techniques, high salt concentrations are not suitable because the salt disturbs the separation in the electric field. So what you do is you apply your sample, and basically you have a sheet of fluid that moves in one direction of liquid. And in this liquid, you have an electrolyte. And either the electrolyte is uniform or it's segmented, or there are several segmented electrolytes. And then you apply an electric field perpendicular to the flow direction of the liquid. And then the liquid flows, and the organelles and the proteins in there, they flow from A to B, from the top to the bottom here, where they are collected. But on their way, due to their charges, they are deflected at different angles. So this electrolyte flow guarantees that they are transported in this electrolyte environment that you can segment according to different pHs, for instance. And the electric field that you apply perpendicularly to that flow direction deflects the organelles, and you collect them at different positions, like in a gradient here. And how that looks like, I show you in this video. Now, this shows you this experimental setup with the computer that is directing it. And the preparation follows in this chamber. The features you see displayed in here. So basically, you perform an isoelectric focusing. You condition the separation chamber with your electrolyte first. You calibrate it. So you get these stripes of the segments. You put the separation medium on top, like shown here. Let's put it again here. Let's look at the isoelectric focusing again, because this is the important part. We condition the separation chamber first with the right buffer and the conditions. Then we calibrate with the electrolyte. Then on top of that comes the separation medium, the buffer that should not have too high salt concentration. And then we load the sample. And before that, we apply the current, and then the isoelectric focusing is established. Then we apply the sample here on one side. And the sample contains, for instance, proteins, protein aggregates of different size, of different charges in this mixture, and they start now flowing through the chamber, and according to the isoelectric points, they get separated and then be collected here on this other side of this gradient where they migrate in little tubes that suck off the medium that comes and transfer it in, for instance, in 96 well plates where the samples can be collected. And finally, the samples end up in these different wells here, like you see it here. So you can use different gradients. You can deplete certain proteins. You can separate isoforms. You can apply a zone electrophoresis with one zone only. You apply a very high voltage here. Then the sample is applied again in liquid in one side, and it can be continuously loaded. And here we have organelles, for instance. These are mitochondria. And these mitochondria can be separated again, depending on how they are charged, how active they are. So you can separate organelles as well, as shown here, and you can collect them. You can even separate entire cells. You can use native conditions, so without detergent. That is also a very important feature. So you don't destroy your sample. You keep it as native as possible here. And this shows you again the setup with the buffer reservoirs, the computer that makes the thing run, and that you can analyze it. So that is free flow electrophoresis, and the chapter is also covered in the literature that you have received for this WeTransfer link. The next technology I would like to introduce is immunoisolation. So this is something rather different. In this technology, we use antibodies And we separate organelles with antibodies according to their antigenic properties rather than their physical properties. So this is something new now. But all techniques I'm showing you now, the free flow electrophoresis, the immunoisolation, the different gradient centrifugations, the density shifts, they all rely on this basic module, how you fractionate cells, how you homogenize them. The quality of your homogenate, the care you take into this first step is then reflected in the quality of what you get out at the end of the day. Therefore, in the lecture and also in the video, in the first video that I've shown you, most of the emphasis was put on the fact how you disrupt your cells to gently release the organelles that you can then purify with this combination of technologies that I'm introducing to you today. And therefore, the second bullet point here reads as follows. This immunoisolation is most efficiently combined with density gradient centrifugation as means for pre-fractionation. And what you do is you use a solid support. This can be a column or can be a bead, where you have antibodies linked to it to pull out organelles which display certain antigens on their surface. And the antigens usually are proteins, can also be sugars, can be lipids. You have a fraction of organelles, and the subfraction of these organelles contain these antigens where you have antibodies against that. And then you have a specific antibody that recognizes this antigen, and you have a second antibody, this linker antibody, we will define what that is, that links this antigen recognizing antibody to a solid support, orients it, and pulls out this organelle fraction. And this technology you can use in different ways. It can be applied directly or indirectly. Directly means you have this little solid support with the three, the linker antibody and the specific antibody, pre-formed already, and you add it to your complex organelle mixture that has been free-fractionated, and you want just to separate out a subfraction of these organelles, and you immobilize these organelles for the antigen, you pull it out, and you bring it to your subsequent analysis. In the indirect methodology, you have this organelle solution and you add the primary antibodies that recognize the antigen, and then you add the beads with the linker antibody and put them in that they bind and you pull them out. So both strategy work. It depends on really on an empiric trying if the direct or indirect procedure works better in your particular case. Sometimes antigens are hidden a bit, and it's easier when the antibodies in the indirect method can freely float through the sample and bind to the antigen. Sometimes it works better when you have these preformed antibody trees already on the beads. These beads are commercially available, as you will see later, and you can buy and use them. Some of them you can make yourself. Now, this binding of the antibody to the antigen is something that needs to be controlled in the experimental conditions. The binding of an antibody to an antigen has a certain time that it takes, and this is what we call the specific binding. So after a certain time, you reach the plateau of the specific binding. The hours are given here at the bottom. And then the specific binding, it cannot be increased anymore. However, the unspecific binding here will increase over time. And at a certain moment, these two curves will cross and you will only increase the unspecific binding. Like with the binding of the antibodies, we need to know that this binding activity is dependent also on the surrounding temperature. I told you already before that everything takes place in a slower way when we decrease temperatures. And the same thing is here. So antibody binding is reduced at low temperatures. With that, even more reduced is the non-specific binding. yeah, because due to the affinities. The specific binding has a high affinity, the unspecific binding a low affinity. And lowering the temperature decreases the affinities in general. The high specific binding will still have a higher affinity. It will take longer time. And the non-specific binding with the low affinity will be largely avoided at low temperature. And this immunoisolation takes place under native conditions, so without detergent. And you might have been confronted with something similar already in your practical lab work, or at least theoretically during your studies, and that is immune precipitation. It's the same principle. For the immune precipitation, we use detergents to solubilize membranes and to pull out receptor complexes, protein complexes that we pull down then with beads, with antibodies for immune precipitation to look, we can immune precipitate the protein, which other proteins are associated with the proteins. We can read that out with mass spectrometry, or we can read it out with Western blotting. And that is the same principle, so that you perform these immune precipitations at 4 degrees Celsius in the cold room. The same will be true for the immunoisolations in the cold room. And you might have wondered in the cold rooms of where you did your lab work during your lab rotations, maybe they have cold rooms at the institute where biochemical work is performed. There you see these rotation wheels, or you see the Western blots shaking at 4 degrees with antibodies in there. It is all made because unspecific binding is lower at 4 degrees. However, it needs to be established because exceeding the time unnecessarily would still increase non-specific binding. So you have to find the right window, the right temperature where you perform your experiment. Some antibodies have such a high specific activity and a high binding activity and affinity that you don't even need to go to the cold room. You can perform in immune isolation even in a few minutes at the bench at room temperature. But these are very rare. Most of them require these temperature shifts and working at 4 degrees that you titrate the conditions. What is important for you to know about the antigen, I explain you on this slide. The epitope must be exclusively present on the surface of the desired compartment. And this is important. I give you a simple example. You are working in a lab where you should immune isolate mitochondria. And the lab leader, the supervisor tells you, yeah, take antibodies against mitochondria, hook them to beads and pull them down. You go to the lab database, you look what is in the freezer, and you find wonderful, highly specific antibodies against mitochondrial matrix proteins. Now, they will not work because the matrix is inside the mitochondria, and antibodies must recognize a protein outside on the surface of the mitochondria. And there you have also to be careful to look which epitope the antibody is recognizing. Is it an epitope that is exposed to the cytoplasmic phase of the mitochondria, or is it an epitope that sits within the membrane space or to the lumen of the mitochondria? Now, they would neither be suitable for that. So make sure that the epitope is exposed to the cytoplasmic space and that you can reach it with the antibody. Of course, in order to be efficient, the epitope must have a certain concentration on the surface, and this concentration should be on the surface of the organelle. Yeah. And immune isolation is easier when the epitope is relatively abundant. The antibodies need to have also to fulfill certain prerequisites. Let's first talk about the linker antibody. Now, this linker antibody binds the specific antibody that recognizes the epitope. Why do we use a linker antibody? It increases the flexibility of the specific antibody because, like for fishing, it makes the cantilever longer and makes it more, more flexible. And it helps you to orient the primary antibody. because with the FC segment, we can immobilize, let's say, the linker antibody in the right direction on the beads already. And then this linker antibody binds then the primary antibody on the FC segment because it's a primary anti-FC antibody. And then the epitope recognizing antibody is oriented into the right direction and can find the epitope more efficiently. Also, the specific antibody needs to fulfill requirements. Not only that the epitope must be on the surface, it must have a certain affinity, so a good KD that is lower than to the -8 usually. The selection of the antibody is more or less an empiric selection. The only real test is the immunoisolation per se. If a monoclonal antibody is better than a polyclonal antibody, and which monoclonal or which polyclonal is better has to be tested. But what can be said in general when you're using polyclonal antibodies, they should be affinity purified monoclonal antibodies because they contain otherwise too many epitope recognizing antibodies in that mixture. But you can enrich the epitope recognizing antibodies by affinity purification of the polyclonal serum that comes usually out of rabbits against the epitope they were raised against a peptide or a recombinant protein. Now, the solid supports that you can use. Here, the composition of the solid supports is a matter of concern. Hydrophobic surfaces are, of course, sticky because they bind lipids. The surfaces should be chemically modifiable, that you can attach antibodies to them. And the solid supports should not aggregate. That means their aggregation behavior should be minimized. When they aggregate already in the absence of cellular extracts, like latex beads, for example, aggregate very easily, then these aggregates, again, like the DNA that leaks out of the nuclei, would generate protein sinks and organelle sinks where unspecifically everything binds in a big clump to it. It should be possible to correctly position the antibody on the solid support, and they should have the possibility that you can purify them by sedimentation at low speed. Around 3000 G and not more, so that the organelles do not co-sediment that have not bound to these particles. Some particles you cannot really sediment at low speeds because they are so light or they have the capacity to float up in every liquid where you put them in. Therefore, the segregation and the sedimentation should be possible. And when they are too heterodispersed, that means they have different sizes, they also have higher potency to aggregate rather than monodispersed particles when they have all the same size. And here are some types that you can use for that. What is used most at the moment are magnetic beads due to several reasons. You can buy them pre-bound to all kinds of different antibodies already. You can pull them out with a magnet on the wall of the tube. You don't need sedimentation by centrifugation. They have therefore a very low background because the surfaces are rather inert. They don't need sedimentation. They are all monodispersed. They have all the same size, and you can buy them in different flavors. The disadvantage is they have a rather low capacity. So this is an analytical method, but not a preparative method like the free flow electrophoresis was that I have shown you, because you can load every hour sample 24 hours a day, 7 days a week, and you can purify and purify with this free flow electrophoresis because it doesn't mind. It runs all the time and you can collect all the time. Here with the magnetic beads, you have a logic end. You can use a certain amount of beads in a certain amount of volume and you cannot scale that up, but it's an analytical method and the downstream methods, the biochemical methods like mass spectrometry have advanced so much that it is sufficient. Other particles that you can buy are latex particles that I mentioned before, or pearl sheet particles. So these are basically like porcelain particles. They have a very high capacity. They are monodisperse. You can hook antibodies on them, and they are commercially available. They have, due to the nature of their surfaces, higher background, and they do not work with all antigens. That has been shown. What is coming from nature, but it's provided commercially, is fixed Staphylococcus aureus cells, which express protein A on their surface. Now, protein A binds antibodies. So you can decorate your organelles with your primary antibody. You add Staphylococcus aureus cells, which have been killed by fixation. Due to the fact that they express protein A on their surface, they can bind the antibodies, and then you can pull it down very elegantly. They have extremely high capacity, surface to volume ratio. They are monodispersed because they are produced by nature. They have all the same size. You can buy them. The disadvantage is you need to separate them with high speed in a centrifuge due to the fact that the surface is a natural surface. It's a bacterial surface. They have some stickage. And the protein analytics becomes a bit difficult because Staphylococcus aureus is a bacterium and has its proteome itself. Now you can subtract the proteome of the bacteria by means of bioinformatics or by identification of the proteins, but you have to be aware of that. And finally, there is something like a fishing net. So not a round surface like a nanoparticle, but a net, a mesh. And these are cellulose fibers. And these fibers you can very easily produce yourself in the lab. You can also buy them. And these fibers have a very high capacity because, like in a mesh or in a net for fishing, you trap everything which has a certain size. And these fibers can also be linked to antibodies. So it's a rather smart approach. It has therefore a high flexibility. You need very low speed to sediment this net plus content, but entrapment background is considerably higher. This shows you again a visualization of such an immunoisolation of endosomes. And all these numbers that have been put here on the proteins in red show late endosomal proteins that have been newly identified and highly specific for that organelle, and the white ones show contaminants that come from the ER, like BIP, chylreticulin, tubulin from the cytoskeleton, actinisoforms. But from just looking visually at the picture, you see this is really as pure as you can get. And there are several thousands of proteins on this gel. When you zoom in with scanners, you can see all of them and you can identify them. But this is as close as you can come to the proteome of a single organelle. Now, finally, I would like to introduce another technology, the last technology for today, and this is fluorescence-activated organelle sorting, or FAOS. This is rather smart because flow cytometry can be adapted to sort and analyze intracellular organelles rather than cells. And you can label these organelles like you label in immunology different lymphocytes carrying different subsets of T-cell or B-cell receptors with different antibodies and analyze them quantitatively, how many subsets you have in which patient under which conditions of the immune response. You can do basically the same thing with intracellular organelles that you label with fluorescent dyes, or you generate cell lines with different colors. And you combine that again with conventional subcellular fractionation techniques. You homogenize your cells according to these protocols I have shown you. You pre-fractionate them with gradients. And then you take that fractions and further separate them, again, instead of immunoisolation, with these features of fluorescently labeled proteins, or even antibodies that are fluorescently labeled, or fusion proteins that carry a fluorescence color, or fluorescent dyes that you have introduced in organelles. And you can label these organelles, mitochondria, Golgi, plasma membrane, phagosomes, endosomes, with fluorescently labeled ligands or with vital dyes. And there are companies out there that supply a large number of these vital dyes. This summarizes the principle of the fluorescent organelle sorting as well as the fluorescent cell sorting for you. So what you have your sample that flows in a buffer and is pumped to a nozzle. And in this nozzle, a hydrodynamic focusing takes place so that that buffer stream is focused and this nozzle is vibrating here. And this buffer stream is disrupted into droplets on that way. And each droplet contains organelles or contains a single cell. So statistically, this is broken up that you have stained cells or stained organelles in droplets and the other droplets that contain nothing. And they pass in that stream for a light source. And this light source is generated by a laser, and it can generate laser light of different wavelengths, and you can read out many different fluorescent colors on the one hand, and you can read out the scattering of the light. So the scattering of the light is how the light is scattered by the size of a cell or by the granularity. So granulocytes have macrophages have a different scatter light. You have a forward light scatter and a side light scatter. All these things in combination with the fluorescence light can be recorded by detectors and sensors and feed it back to a computer device that coordinates everything what takes place here. So now you can give the computer and the instrument the command deflect everything by adding a charge here. So the red vesicles would be negatively charged. The green vesicles, for instance, would be positively charged here at the end. And then you have an electric field that is generated by two electrodes, a plus and a minus electrodes. And these vesicles that are generated by this vibrating nozzle have a different charge being put on them, either if they are green, red, pink, magenta, whatever you want, which features, and they are deflected differently and can be collected. Here, for example, the red ones are deflected to the plus electrode, the green ones are deflected to the minus electrode, and what is collected in the middle is the waste, which contains neither of these colors. So by that, you can generate very, very pure organelle fractions. When you take into account some of the criteria, because for organelle sorting, the sensitivity is obviously a major concern because the organelles are smaller than cells, and intracellular organelles therefore have only a limited number of dye molecules associated with them. That means besides the physical properties of the dyes, absorption coefficient, quantum efficiency, and these physical considerations, background signals coming from autofluorescence can be a critical limitation. So why do I mention that separately here? Certain amino acids are autofluorescent. And you might have observed that when you look at cells in a microscope, in an epifluorescing microscope, and you look at the negative control that should not contain any antibody, or any secondary antibody, and should be neither green or red, and you shine the fluorescence light on them, and you read out in the green channel or in the red channel, and your negative control all of a sudden has a background of fluorescence. And some cells are brighter and some cells are very dim and you hardly see them and you are confused and you don't know what is it about. Well, this is autofluorescence coming from living cells that produce amino acids, and some of these amino acids are autofluorescence. Just keep that in mind. And therefore, these different metabolic states with the production of different amino acids can cause autofluorescence. So for which organelles can you use that? Again, of course, endosomes, because you access them from outside and can load them transiently with fluorescent membrane dyes or fluorescently labeled ligands under different conditions. But you have many cell-penetrating live cell dyes available. There are dyes, for instance, that that incorporate into mitochondria like rhodamine 123 only when the membrane potential is intact, or they shift in color when the membrane potential is not intact. So you can sort active mitochondria from inactive mitochondria, and many, many tricks more you can play. And you can generate fusion proteins with GFP, YFP, and use them as organelle markers. And again, the visualization, you know, the starting fraction of the PNS on the 2D gel, the organelle enriched fractions. You see how you enrich these proteins that you don't see here. You see on the right-hand side, and these proteins. So these are proteins you would never have seen in the total cell lysis, or you would never have seen in the starting fraction. So these are very high-end techniques and powerful techniques that you can use. And that is what people have then used to make organelle proteomes, Golgi, plastids, implants, nucleus, peroxisome, ER, and they put all of these organelle proteomes together in order to understand the human proteome in its entire entity. You can combine that with protein correlation profiling, where in a gradient which proteins migrate under which conditions. You can use principal component analysis, like for gene expression analysis, and functionally filtrate protein components out of that. This is given a bit in the literature that I've accompanied for you. You can generate a virtual phagosome map and basically map all proteins, all signaling proteins on a single organelle, like a phagosome that is an organelle that is used for getting rid of phagocytosed material, bacteria or cell debris or autophagosomes in the cell. And you can start to learn more about these organelles and their involvement and essential biological functions by learning about their proteomes. You can use it also for lipid distribution profiling, for lipid profiling, like it is shown here. So we can analyze the lipids of organelles under different starvation conditions, under different treatment conditions upon medications. So there is no real limitation of that technology. And that brings me now to the end of my lecture. And here I would, before I explain you the homework, I would like to ask you if you have questions to what I have told you. Are you still there?
Yes. Yes.
Okay. So, maybe I explain you what I expect from you to do now. The homework is as follows. You should write a short essay on subcellular fractionation. The choice of the content and the topic is up to you. You can write a summary of what I've told you today. You can pick out one certain technology and describe it, and you can do your own literature search and add a few examples or what you find to be interesting there, how it has been used. You can reflect on your own work on the bench that you did and where something like this could become interesting. The entire thing should not be more than 4 pages or so. And what is important for me is that you respect the general rules for scientific writing and scientific work. No cut, copy, paste from existing literature. Try to write it in your own words. You know, one main part of my work as a scientist is that I write papers, that I write reviews, that I correct the master thesis, bachelor thesis, diploma thesis, PhD thesis of my students. And I see immediately if you cut, copy, paste text. And I don't want you to do that. I want you to use your own words in English. And if your English is bad, it will improve by doing that. It's an exercise for you. You have to write a text in 4 pages. You have to put in citations. You can take pictures from the literature, from the reviews I've sent you. You have to cite it properly. You write an introduction, a kind of abstract, a title. your name, your matricle number, your study number, then a headline, what is it about, a short summary, an introduction, or the text, and you cite the text properly, you put the literature at the end. The figures you take have to be cited back when they have not been drawn by yourself. You can take every figure out of the literature, but you need a proper citation, no cut, copy, paste. You can use the online material supplied by me, and you can complement it with your own searches. And you send this text to me, and I promise I will read everything. And I will comment on it. I will either praise you and say it's perfect, or I will give you tips and hints what you have overlooked, or when the English needs some adjustment, I will help you with that and send it to you. But this exercise will be rewarded for me that you send me a good homework, and will be rewarded for you that you get a good mark at the end of the day and get it for your study curriculum. And this should be completed in due time. Yeah, so that we need to agree now on a date where you will be able to complete that. This can be at the end of the semester, that can be in the middle of the semester, that can be rather soon. My recommendation is, if I would be you, I would write it rather soon and send it back to me, because then it's done. You have it rather fresh in mind. I have put here in the back the literature, so you have what I have sent you, isolation protocols, general articles about fractionation with detailed protocols, an article about organelle proteomics, another article about subcellular fractionation, electromigration analysis and mapping of organelles, an article of organelle omics, reviving strategy to untangle the complexity of cells. So you find a lot of things here that you could use for your homework. But now we have to agree for a date where I close then the submission time and where I correct it. And you send it to me by email. And this is my email address, and also the colleague, I think it was Emma Steiner, who had problems with downloading the PDF file. She can use this email address to contact me, or when you need any other material, all of you can contact me at any time with this email address. When you contact me and you send me back your essays, when I receive it, I usually reply immediately or within 24 hours that I have received it and everything is okay. When I don't reply, something has happened, because I'm reliable and I always reply when students write to me. So when I don't reply to me, don't be shy and send me immediately the next day or the day after a reminder if I have received it or not. Things can happen to my mailbox that it overflows and I lose correspondence, or sometimes things end up in the junk folder or in the spam folder without my intention that they end up there. And then I might lose them. I regularly control that. But sometimes when there are attachments on emails, when they're rather large, the spam filter university removes them. But this should not happen in general. But when I don't reply immediately with an okay, or I received it, then something has happened, so remind me. And then I will look at it immediately, will start correcting it, or will approving it, and I send you back the mark. And at the end, when I have collected everything, I contact the study section, and Renate Fauland will give in the marks then for me, and you will have that rather early when you need it for stipends or anything else. So now it's up to you that you make a proposal until when you send that back, that we can fill that in. Please speak up now.
What do you think about the 15th of April? Because I checked when the lab courses are.
Yeah.
Most of them are from March 28th until April 8th. So whoever doesn't finish it before, I guess many of us will have to attend lab courses.
You know, for me, this is perfectly fine. So Andrea Lawrence suggests middle of April. Emma Steiner asks, what's about the end of April? Are there more voices?
End of April is fine with me too.
Yeah.
Is there anyone who needs the grade early?
Yeah, because if anybody needs the grade earlier, then we have to put the deadline earlier. Because you also have to understand that I have other teaching obligations and scientific obligations and committee applications and university applications. So I lose then attention when the things are too far away. Okay, so end of April is after Easter. Easter is 17th, I think, of April is Easter. Yeah. So then we agree, we make it end of April.
So 30th of April or?
Yeah. So April 30. Let me have a look. Oh God, that had collapsed now. April 30, I scroll to the bottom here. Wait a second, I mount it again in a second. So this is binding now for everybody, April 30, 2022. And this is where you have to submit it to my email address.